welcome Marcus Roper here to our seminar series and introduce him. Marcus you're an associate professor now, right? Or you will be soon.
Starting July, yeah.
Yes, you're an almost associate professor here in the Department of Mathematics on campus. And I should have kept my glasses on now. I can see how much I can remember. I'm still in denial about glasses. Marcus got his PhD at Harvard in 2007, and then he did a postdoc at Harvard, and that was followed by a Miller postdoc at Berkeley. And he arrived at UCLA in 2011 and got a Sloan Fellowship, which is a prestigious award, which is great. And he is an applied mathematician who has a nice biological bent, which is why he's here speaking today. And he does things like bioballistics. That's in part of the title of his dissertation, which I can't read now because I don't have my glasses, but I like the word bioballistics. And beautiful phrase, mushroom wins. I mean, someone should write a book entitled that, right? And today he has done a lot of work on fungal spores and how they move around, and modeling, mathematical modeling of such, and worked with Anne Pringle, who we have an outstanding offer for at the moment. And today, he's going to talk about myofluidics, mushroom winds, and communicating on fungal freeways. Thanks.
Okay, thank you. So I'm really stoked to have the opportunity to try talking about some ideas that are very, very recent, coming out of the work that I've been doing. So a lot of the ideas, I think I'd be especially appreciative of feedback. I mean, if you have I don't know what the culture is in biology talks, but if you have an objection, just holler. It's you might be right, okay. So what do I want to do? The first thing, if I'm too quiet at the back, tell me, because I have a tendency to taper down when I get excited. My voice goes down. So okay. So what do we study, and why are we interested in it? And I don't know. The next few slides might be completely specious for this audience of learned biologists. But for me, they were very interesting, and so I just present them as sort of part of my personal trajectory to being very interested in studying fungal biology. And so,
it won't leave you alone.
How on earth?
You turn off your Wi-Fi.
I don't know where my icon is anymore. Okay, thanks. Okay, very persistent, very, very helpful. Okay, so this is a game you can play, and I'm sure, like, as biologists, you'd be much better at this game than I was when I played it, which is you start from a fact, and there's a lot of controversy about this fact, and there's a lot of big uncertainty, big error bars on this number, but there is some consensus, and the consensus is sort of converging to within an order of 10 on the number of species that there probably are. If you're only interested in multicellular species, if you're interested in the bacteria and things like this, I mean, I don't know. Nobody has any idea how many of those species there are. But the general consensus seems to be that there are about 10 million multicellular species on Earth. And to me, as a mathematician who knows very little biology, this is kind of like a fun exercise to start seeing whether you know where those species are or what they are. So you start by trying to name the species. And so the first, I don't know, don't worry, we won't be trying to name all 10 million, otherwise we'll be here for weeks. But this is sort of, so I start and I name the first species I knew of, which is humans, which is everyone, you, me, our kids, our neighbors, that's us. So we're one of 10 million species. And so really sort of like we're the razor edge of like diversity on Earth. And so then you start seeing if you can fill out the rest of this pie chart with the other species. And clearly if you name individual species, I mean, you're not going to get to 10 million. Well, at least certainly not on any reasonable time scale. So let's just name clades of species. Clades, phyla, whatever you want. So let's start by considering simply the mammals. Well, there are 5,000 species of mammals. I don't know, there's lots of interest in the mammals, but there's still like this tiny slice, this tiny razor edge of a pie slice of this global diversity. So then you just name a whole bunch of clades. So you'll name the mammals, fish, birds, reptiles. Well, that's a lot of species in those groups. And that brings you up to 55,000 species. So it's a lot of very interesting and charismatic species, but still only a razor edge of diversity. Okay, and so then you sort of, well, you go, oh, plants, there are lots of plants. So, well, yeah, in some sense, there are a lot of plants. There are 300,000 species of plants. That's that's an immense number. But still just a tiny, tiny slice of the pie. So what's left? Well, what are all of these other species that aren't in these charismatic groups? Well, this is kind of, to me, and maybe this is sort of my overreaching in saying this, but I kind of feel like, from the point of view of someone who's just learning biology, this is like dark matter. This is the biology you don't learn about in high school when you take those biology classes. So what is the dark matter? Well, a lot of it is fungi. Not all of it. There's a lot more dark matter, but there's still a huge chunk of this dark matter is the fungi. And so the general consensus is that there are 3.5 million species of fungi. These are very, very understudied. So this is just kind of how we started, how I started to think, well, maybe there are stories, there are so many interesting stories in this section of the pie, and maybe there are also interesting stories here. So this talk is going to be about a couple of just two recent interesting stories from many that we're just discovering in my group. But I guess, this is the last thing, I don't know, this is like beat you with a stick slide. Why should you care about the fungi? Well, maybe the fungi care about you. I mean, maybe they're quite capable of wiping out an organism that you do care about. So if you go hiking in Kings Canyon right now, and this is a photo from Science magazine, you'll see this sort of complete destruction, annihilation of the amphibians across America, which has been caused by a fungal disease called chytridiomycosis. And this is so they keep hammering home the point slide. There are so many emerging diseases right now caused by fungi that really peril our ability to produce our food. They imperil native populations of corals, forests, amphibians. And so there's certainly a reason to care about the fungi, even if your main interest, your dominant interest is in the other wedge of life. Okay, so this is my beat you over the head slide. We're going to move swiftly away from this because I hate this slide. I hate the portrayal of fungi as these sort of nefarious organisms. So the stories that I want to tell involve how fungi can be heroes of a story, how they can solve difficult problems and achieve very, very challenging tasks. Okay, and so we've coined a name for this, and it's a punning name, so I apologize, but the name is mycofluidics. And what does it mean? It means that molds solve physical problems. And so the physical problems that I'm going to talk about today are connected with the problem of dispersal, how you get around if you're a fungus, and also a problem involving growth. And so we'll talk about that growth problem in a little bit. So the first problem is about dispersal. I want to acknowledge my collaborator, Emily Dresser, who is an engineering professor at Trinity College. Okay, so what is the general problem of dispersal? Oh, my video is aren't synced. So fungi disperse using spores. Okay, and these videos are showing, and hopefully we can just play these in a rational order. So lots of things disperse using seeds and spores. It's a very common strategy. You sort of create something and then you fling it out as far as you can get it to go, and hopefully that propagule will then seed a new organism. Okay, and so this is very, very, you see this in many different groups of organisms. You see it in plants, you see it in the fungi. The problem is the scaling of this process with size. And so this is a problem that was first identified by Steve Vogel in a paper where he actually coined the term bioprojectile. And so the contrast is shown very clearly if you consider dispersal of seeds. So this is, I don't think this is jewelweed, but it's related to jewelweed. It's sort of in that group of plants. And so this uses a ballistic mechanism to eject seeds, and the seeds are a few millimeters in size, and they're ejected at a speed of 4 meters a second. Okay? And so we compared this with a fungus, and this fungus also uses a ballistic mechanism. It's a different mechanism, and we're not going to talk about the details of it. It was sort of a lot of work has been done by people like Anne Pringle to understand the physics of how these spores are being ejected. But the important thing is the contrast in size. These seeds are 3 millimeters. These spores are 5 microns. The speeds are about the same. It's about a matched speed of 4 meters a second. Okay, and so it's hard to see this from the high-speed movies. The high-speed movies just show the glory of, okay, so that's the spore being ejected. It does not sync. This one is supposed to play first. So, Okay, so now you'll see this one being ejected. Okay. So different processes, different scales. Well, how far do these seeds go? These seeds go about 0.5 meter. So you get flung some significant distance from your parent organism. How far do the spores go? The spores go 0.2 millimeters. Why do they not go very far? Well, you have to think about the way that inertia scales with size. So what keeps you going as an organism, as a projectile, is your mass, your inertia. And so your inertia depends on your volume. And so if you sort of think about how your volume scales with your size, if you halve the size of a projectile, you're going to reduce its inertia by a factor of 8. Okay, and so there's a little bit more math that you have to do, but effectively that means that you reduce your range by a factor of 4. So small objects are very, very ineffective as projectiles. And the boundary seems to be about the boundary between seeds of plants and spores of fungi. These are terrible projectiles. This goes only 0.2 millimeters. This goes close to 1 meter. Okay, so people have known about this for a while. They might not have couched it in these physical terms, but it's generally being understood that the initial impulse that a fungus is capable of imparting to its spores won't carry those spores very far. So that's okay. I mean, the spores are very small, they're very light, they can be carried away by the wind. And so the prevailing view is summarized in this quote, which I couldn't really think of a way of condensing it, but this is sort of the sort of view that comes actually from the fungal biology community, from the phytopathology community. And this idea is that birds and mammals, the way that they move across landscapes is very sophisticated. They're very smart. They do the right things. By contrast, fungi are just terrible. I mean, they have no, they're just awful at dispersal. I mean, all they can hope to do is to produce enormous numbers of spores, most of which will end up on uncongenial environments. So you just make as many spores as you can, and you just hope that the wind will catch some of those spores and carry them to a new place for you to settle. Okay, and this actually comes from the fungal biology community. This is not, this consensus has been around for a long time. So we sort of wondered, and I'm going to give you some context for why we've started to wonder this. We sort of wondered whether, I mean, if all you have to do is produce, millions and millions of spores, why is there so much fungal diversity? I mean, why is there so much diversity in the food bodies, the organs that are being used to produce and liberate the spores? Why are there so many different solutions to a problem which is just a very simple problem? Make a lot of spores and hope. Make a lot of spores and cross your fingers. And so the first inkling that we had, and this is not what the subject of this talk, but the first inkling that we had that the fungi might enjoy more control over their dispersal fate than this sort of trammeled up in this phrase that I just showed you. So it comes from this very old observation. And this has been known about since the 18th century. And so you'll see sort of old pictures like this in a textbook, which is that some fungi, they're called the discomycetes, they liberate a lot of spores at once. They synchronize their spore ejection. They make lots and lots of spores, and they release them as quickly as they possibly can. Okay, and so what happens when you look at the spores being ejected? Well, I went to Cornell, and I took videos where they grow these fungi, because it turns out it's an important pathogen of crops. So one of these species is anyway, and I sort of took videos of the spores being ejected. Okay, and so here is, I don't know if any of my videos are going to play. Okay, it's going to play. So this video has been slowed down by a factor of 5. You'll see the spores being ejected here. Like this is really a proper-sized petri dish, about 10 centimeters in size, and that cloud that you're seeing is the cloud of spores that's being released in this sort of synchronous, coordinated way. Okay, and so dissecting these dynamics really started a beautiful collaboration with Agnese Seminar, who's actually sitting over there, and with Anne Fingal, who's already been mentioned in this in Blair's introduction. And so what did we learn about these dynamics? What is the benefit of ejecting a bunch of spores at once? Well, you can see from the video the spores go a lot farther than 0.2 millimeters. They're going 20, 30 centimeters. There's a huge range enhancement. So what is the mechanism for this range enhancement? Well, a single spore, it's ejected, it comes to rest, but its momentum has to go somewhere. the momentum that the spore is carrying doesn't just dissipate, it has to be conserved. That's sort of one of our physics 6 principles. So where does the momentum go? Well, the momentum has to go to the air. So in coming to rest, the spore sets a tiny packet of air into motion. Well, a single spore can't move a lot of air. But if you have 100,000 spores, and they're all being ejected together in this synchronous way, they can move a lot of air. And so we explored this, well, Agnesi explored it using direct numerical simulations of the airflow created by these spores. And you can actually investigate it in one of her simulations by plotting the speed of this surrounding airflow, and that's this sort of red curve here, on cross sections at different heights of the jet, and the speed of the spores. Okay, and so the black curve here is the speed of the spores. What you see is as you move up from higher and higher and higher into the jet, the spores decelerate, but the air around them accelerates. And so until at some point, at some critical height in the jet, the spores and the air are moving at the same speed. And so then there's no drag on the spores at all. The spores have stopped moving the air, and the air is now pulling on the spores. So this sort of started with us. So this is not the subject of our current talk, but this started just as a general line of inquiry about whether fungi had to live with the environments that they were given. So here you have a fungus that isn't able to project its spores very far into still air. Its remedy is to create an airflow, and that airflow disperses the spores many-fold further than they would go in still air. Okay, and so we sort of wondered, I mean, this is one group of organisms. Are there other, the rest of this huge chunk of life, are there other mechanisms that fungi are using in order to manipulate or create airflows? And so there were a few mysteries that got us started interested in mushroom forming fungi specifically. So this is a different phylum from the one that I talked about with the puffing and the synchronized ejection. These are the Basidiomycota. And so these mushrooms, there are a few curious observations about them, and I think that they're all connected in a single story. The first thing is, if you go out in nature, so the mushrooms sort of show up, the mushroom is the organ of dispersal, and so the mushrooms sort of show up wherever they can, in the sense that if there's a fungus that's growing in a tree trunk, there are a few interstices in the bark that the mushrooms can show up in. So you'll tend to see a lot of mushrooms showing up in the same place. They'll grow crowded on a log. The other place where you often find mushrooms is very close to the ground. Now remember, our prevailing view is that the spores have almost no ballistic capacity. They don't go anywhere but by their own initial impulse. What carries them is the wind. Well, this is a terrible way to grow your spore dispersing mechanism if you want your spores to be caught by the wind. There are these tiny, tiny gaps here, or you're growing very close to the ground. Measured wind speeds in these kinds of environments are less than a centimeter a second. So it's not really very good for dispersing your spores. The second observation is, so there's this I don't know if anyone who goes mushroom hunting like I do. Yeah, who has this book? This book is awesome. I really love this book. And so this is the observation. I wanted to use it as the dogma among mushroom hunters, and it's a true dogma. Many dogmas are true, which is that the time to go mushroom hunting is about 3 days after it rains. In Southern California, you might go out 1 or 2 days after it rains, but this is sort of the general consensus we have. Mushrooms depend on rain; they depend on water to be created, and so there's sort of lots of reasons for that. You could say, well, the mushroom is actually a sort of hydraulically inflated structure and things like this. And so the mystery we had, and we puzzled over, is if the mushrooms depend on water, you would expect that they would do a fairly good job of conserving it. And so this is sort of my observation, not really being a biologist, and so sort of I apologize if the language I'm using is loaded here. But this is this observation, and we who cook with mushrooms know this. If you stick a mushroom out overnight in your kitchen, it dries to a husk. They lose their water very, very quickly. So you watch mushrooms over the course of a few hours, and they just basically disappear. So if water is important to you, why don't you do a good job of conserving it? Why don't you do a good job of stopping it, just evaporating away? And so we actually sort of tried to put some context on this observation, and this is sort of our honest effort. And I apologize if this is a physics experiment applied to a biology process. But we tried to see, you know, what the rates of evaporation were for the fungi and how they for mushrooms and how they compared to other other tissues and things like this. And so what we found is that this is two different groups of mushrooms. They tend to lose water at a rate of like 4% per hour of their body mass per hour. And so if you compare that with sort of what we would sort of in the lab use as a mechanical analog of a tissue, which would be a hydrogel, so just basically a lump of agar, it's about equal. So wet things tend to lose water at about this rate if they have no adaptations whatsoever. I mean, a lump of agar is not trying to conserve its water. It's not trying to do anything at all. It's just there, you made it. And so then we thought, well, to try and put some context on this, let's compare it with some other tissues. So we took some chicken muscles, some chicken breasts from Rayleigh's, and we sort of we just sort of saw how quickly that dried out. And actually that did a better job of conserving its water than the mushrooms did. I mean, chicken breast isn't designed, it didn't have skin on it or anything. I mean, it's just sort of a lump of chicken you would expect. You know, it has no particular need to conserve its water because the epidermis of the chicken is what is involved in what makes the barrier against water loss. So the other thing we compared it with were plant leaves. And so plant leaves have deliberate mechanisms for controlling their water loss. I'm not an expert on plants, so I might only be naming one of them. But one thing that plants can do is they can close the little pores, the stomata, that are involved in allowing water to escape. And with the stomata open, just the big waxy cuticle on the plant leaf means that it loses water significantly more slowly than the mushrooms, although it doesn't do as well as the chicken breast. But then once the stomata close, the rate of water loss just plummets. So there are adaptations that exist that can be used to conserve water. You know, so why do mushrooms not have those adaptations? Well, I don't know. I mean, I like to credit the mushrooms as the hero of the story. Well, my claim and my hypothesis is they don't try and conserve water because they want to lose water. So this is and I realize I'm not a biologist, so using words like want is very loaded, but this is the language that we use, and so this is where this story started. So I come from a sort of engineering and physics background, and so one of the things one of the nice stories involving the early engineering of flight involves this guy called Arthur Cayley, and so Arthur Cayley was one of the first pioneers of the science that became aeronautics, and so he was one of the first people to try and consider how we might achieve flight. And so he had a very specific idea, and his specific idea was that we should use water vapor. So we should fill big balloons with water vapor, and then they will float up, up, up. So why could water vapor be used to get balloons to float? Well, water vapor, if you sort of look at the size of the molecules, water vapor is made up of H2O. And H2O is a very small molecule. Air is made up of N2. N2 is a bigger molecule. So water vapor is actually lighter than air. So in producing this huge amount of water vapor, you're actually making a buoyant gas. And so I thought well, maybe that's involved in dispersal, and maybe that's helpful to you. So the story turns out to be a little more complicated than that, but this was our starting idea. And incidentally, nobody ever got a balloon to take off using water vapor. I mean, they're actually still trying. I mean, so God bless them for so there's a community of people who are still trying to sort of realize Arthur Cayley's dream, but it's not happening. But they only had 200 years to sort of get this to work, and clearly fungi have been around for a billion years. I mean, we know from the fossil record. So there's a sort of a good sense, a good indication that maybe if there were a way to engineer something using water vapor, they would have achieved it in the time scale that they've had. Okay, so one more observation about water vapor, and this is very familiar to you if you sort of go up to Sunset Canyon Pool, and you take a few laps of Sunset Canyon Pool, or one lap of Sunset Canyon Pool if you don't swim very well like I do, and you get out, and immediately you start to feel cold. And so there's another effect connected with this, and that is that evaporation also has a cooling effect. And so this is sort of engineered into lots of devices that we use around, like things like refrigerators and swamp coolers and stuff like that actually connect with this. And so the idea is if you evaporate something, you have to give it a little bit of energy. There's an enthalpic barrier that you have to cross, going from your liquid phase to your vapor phase. And so energy has to be given to to the thing that's evaporating, and that cools whatever body that water is clinging to. And so this is also a significant effect, and we had to consider it. We had to look for it. And so this is actually so we looked at to see whether the mushrooms were creating significant cooling of the air around them by doing this process of evaporation. And so we made some measurements, and we found that typically the order of temperature difference, and there were two different measurements here. We sort of had some mushrooms outside and some mushrooms in the lab that we put in boxes, because this is where we can control the airflow. And putting something in a box reduces its ability to reduce temperature, because you sort of reach a thermal and vapor equilibrium. And what we found is that there's this temperature difference of about 1 degree, up to as many as 5 degrees actually, temperature difference with the surroundings. And this has actually been measured before by some other people who looked at mushrooms, although they made no physical attribution to why the mushrooms, what functional benefit there might be for the mushrooms to be cool. So there's also a temperature difference. Well, temperature also changes buoyancy. So water vapor lifts things. If water vapor is buoyant. Temperature, if you cool something, it becomes denser. And denser things tend to sink. So your water vapor trying to lift, cooling trying to sink. And so we have to compare those two effects. I apologize. We have some math, and so we'll just sort of gloss the math, because I don't think it's hugely important. I mean, I know that you guys can do math, but I don't regard this math as terribly important. We can do some sort of simple estimates of which of those two effects is most important. So if you want to know the effect of the water vapor, well, you have to calculate the partial pressures of the dry part of the air and the wet part of the air. You equate them, and from that you can calculate the density, using the ideal gas law. And it turns out that the water vapor has reduces the density of the air by about 2 times 10 to the minus 3. Okay? And then we also have to contrast that with the cooling. Well, cooling densifies the air. Again, we can use the ideal gas law to calculate how much the densification of the air is. And it turns out that the cooling effect is 4 times 10 to the minus 3. So note the sign difference. Okay, and so this is bigger than water vapor. So the cooling effect should be dominant. And so remember, cooling is sinking. Well, is sinking useful for dispersal? Well, I mean, if something is sinking, it also tends to spread out. Think about pouring a bucket of water on your kitchen floor. I mean, gravity is what pulls the water down, but in order to get as low as possible, it also spreads. So we wondered if there might be some spreading effect from having a cool, dense air. And so we did 2 very simple experiments. I mean, if I'm not a biologist, I'm also not an experimentalist. So my experiments may be regarded as embarrassingly simple. But here are the experiments. The first thing we did is we just balanced mushrooms on stilts, and we looked to see how far the spores got. And so we look at, you can maybe even see the spore deposit under this mushroom. So we sort of measure how far the spores can get. We're engineering this very small gap, which remember is replicative of what happens in nature, where the fungi go very close to the ground or very close to each other. The other experiment which we did is we use a laser beam, so we create a laser sheet, and in that way, as when spores pass through a laser sheet, they scatter light and they appear as bright specks. And so we needed to have a very sensitive way of capturing that light, so we used a high-speed camera to do that. Okay, and so what did we see? Well, this is sort of two ways of looking at this. We can measure how far the spores go. We can also look at the light that we're seeing. And so what you see here is a sort of a mushroom. And so this is sort of just to orient you. This is what a sort of still frame from one of our high-speed camera videos. And you can see that the mushroom, it's sitting above this small gap, and the spores are being forced out from under that gap by the cooling effect, we think. We'll do a little bit of math to verify that. And we can measure how dispersal distance depends on height. And it turns out this black line here is a 3 to the 2 power law fit. And so we'll explain where that comes from in a little bit. Okay, and so there's some questions you might have whenever you run an experiment like that, which is that the speeds of flow that we're seeing are just a few centimeters a second. They're consistent, which is great, because they keep the spores, a speed of a few centimeters a second is enough to carry the spores with it, because the spores sediment very, very slowly under their own mass. Remember, they're very, very small. But you might wonder, are there any confounding effects? Are we just sort of measuring the convection of the lab? So a very simple experiment you can do to check that is you put your mushroom cap in a box, and you measure the distance that the spores disperse from under the mushroom cap. Okay, and so then you take a print on a transparency, go away and photograph it. Then you do exactly the same experiment, but you turn the mushroom round. So the mushroom has been turned around relative to the lab, but the box has the same orientation. All of the currents that are present in the lab should still be there and pointing in the same direction. And what we found is that the spore dispersal pattern that we see in these scenarios, so this in the lab frame would be rotated relative to this. The currents that we see have the same orientation relative to the cap. So that if you turn the cap around, the current also turns around. So this is a very basic test that we're actually being careful enough when we're measuring what the airflows that the mushroom cap itself is capable of creating. Okay, they're not identical, but they look very similar. Okay, and just sort of one of these, you spend hours in the lab taking videos and you just want to show them to people. So we sort of have some videos of our slow. This is obviously taken with a high-speed camera, so the actual thing that you're seeing here, I think, is slowed down by a factor of 4. But okay, so this is the math part, and this is where it gets interesting. So in general, even when I'm talking to like a sort of audience of pure mathematicians, I don't really like to talk about math because it's sort of complicated, and I don't know. I mean, you just sort of feel like you're just bludgeoning people by telling them what you spent your time doing rather than illustrating something. So I want to talk about the math of this, but I really just want to be talking about the physics. So we'll show some estimates, and then I'll show how these make up a physical story. Okay, and so the natural question is, how far and how fast can you disperse your spores by creating these density differences? So initially, we'll just consider the fact that the mushroom is creating cool air that's spreading. So we ignore the buoyancy effect because the buoyancy effect is smaller. Okay, and so there's an estimate that you can do, and it comes from classical fluid mechanics, and we won't worry too much where it comes from, but it sort of comes from balancing the inertia of the spreading current against the buoyancy force that's trying to get it to spread, and from that you can estimate the speed at which the current should spread. Okay, and so the current will spread at a certain speed. And how far will you go in that current? Well, you're also sedimenting. Gravity is pulling you down to the ground. And so the time it takes you to fall from the top of the current to the bottom of the current is going to be just the distance you have to fall, which is the gap height, divided by the speed at which you fall. And so we can estimate that, and it turns out that you have about 3 seconds. Each spore experiences the gravity current, or the cold air current, for about 3 seconds. And so from that, you can sort of put these things together, and you can see how far it should go. And it turns out the distance it should travel, ignore all of the other terms in this, but scales like the gap height raised to the 3/2 power, which was the same scaling as our real data shows. And actually, so in an engineering talk, you can't show data like this. There's like so much scatter. But you guys are biologists. We're the community of biologists. We understand. We understand what data looks like. And so this is actually sort of reasonably in favor of the 3/2 scaling law. But there's another thing, which is sort of we can actually ask, what is the capability of this organism to disperse its spores? So remember this 3 seconds. You're in the gravity current for 3 seconds. So you're in this cool current of air for about 3 seconds. Well, this cool layer of air that's spreading is also being exposed to the ambient, which is surrounding the mushroom, and that's warmer than the mushroom is. So it's also warming up as it spreads. So the gravity current, or this cool air current, is spreading out, and it's warming up. How long does it take it to warm up? Well, again, we can use our physics 6 argument. Actually, I don't know whether this is taught in physics 6, but we can use our basic arguments from sort of considering how much energy the air can store and how quickly it loses that energy. And we can do a calculation that just sort of involves the specific heat in this cold gravity current and the rate at which it loses specific heat, which is connected to this number, which is a coefficient of cooling. And it turns out that it should warm up in about 1 second. So it doesn't. These numbers aren't very easy to reconcile. You're in the cool air current for 3 seconds, but the cool air current stops being cool after 1 second. Okay, so the cool air current actually goes away, and now what's happening? What are the dynamics that are going on here? Well, there is another buoyancy effect. This is the water vapor. Water vapor isn't lost anywhere near as quickly as heat is lost. So you're still in wet air, even though that wet air isn't cool anymore. In fact, the wet air is buoyant. Well, there's a little piece of sort of physics. This is a very, very classical problem in physics that dates back to Lord Rayleigh. And this is this idea that the cool air current is now a current of wet, buoyant air. And so it wants to rise. It wants to rise now that it's lost all of its coolness that was keeping it on the ground. And so what stops it from rising? Well, what stops it from rising is that it's in contact with the ground. And there are viscous stresses that come from being in contact in the ground. The air itself is sticky, and it will tend to stick to the ground. And it has to overcome that stickiness if it wants to rise. And so there's a calculation that goes back to Lord Rayleigh about how buoyant you have to be in order to overcome the stickiness of the ground. And that comes down to a dimensionless number, which is called the Rayleigh number. And the Rayleigh number expresses the ratio of the stickiness of the air on the bottom to, on the top, the buoyancy force that's trying to lift it off from the ground. And the other thing that shows up is the diffusion constant, how quickly water vapor is just lost by diffusion. Okay, and so the Rayleigh number here can be used as a threshold to determine at what point the water vapor is capable of lifting the current, the spores off the ground. And it turns out that the critical number, and there's a detailed calculation you have to do, but the critical number is on the order of 1,000. So bad news, when we plug in our numbers from the real experiment that we're working with, our Rayleigh number is a 10-fold too small. So the buoyant air isn't capable of lifting the spores from the ground. Okay, but it still will cause them to spread. So the old scaling still works, you just have to change the density factor that's involved in there. So that's why we continue to see this sort of h over 3 over 2. Well, this is sort of like could almost be a physics talk at this point, but there's one extra interesting facet to this, which is that the ground is not flat. So this is let us agree on this. The ground is not flat. In nature, the ground is very seldom flat. And particularly for these mushrooms that are all crowded together with each other, like you don't get to travel far before you hit another mushroom. So what does that mean? Well, remember this spreading current can spread along the ground. It's buoyant, so it doesn't actually care if the ground is sort of vertical. So we simulated in a sort of our experimental kludgy way the effect of there being another mushroom next to the one whose current we were looking at. And okay, hopefully this video will play. So this is the mushroom. This is the gravity current that it produces, the cool air. The cool air is now warm about this point, about 1 second out, and it's buoyant. It can't climb under its own, but as soon as it hits this barrier, as soon as there's something crowded in with it, it can go aloft. So this mechanism, although it doesn't create a wind in it's a full stop, if it's combined with crowding, which we used to think was bad for dispersal, it can actually lift the spores a long distance aloft. So the mushroom is engineering an environment that's helped by crowding that helps its spore dispersal. And so although we think about the mushroom as being sort of an organ for producing spores, in a very real sense, it's really an organ for producing water vapor. It's very engineered in a very sophisticated way to create its own dispersive convective wind. Okay, so story number 1. I guess I don't have time for the full story number 2, but this is I like to tell this story in LA. So this is could you get to work any better? This is basically, I mean, what's the best way to get to work? And so it's connected to another aspect of fungi. And so I said that this is probably known to you guys. A mushroom is not a fungus. A mushroom is just an organ that a fungus uses to disperse. So what is a fungus really? Well, a fungus in general is really just a network. So if you look at one of these fungi growing, the network, you see this very dense, branched, interconnected network of tubes. The tubes are called hyphae. And so we'll be referring to them as that. And so what's the function of this network? Well, one of its functions is to balance flow and growth. So the way that the fungus grows, the way that the network expands, is by tip extension, which is continually, if you look at one of these hyphal tubes, you see a continual growth of the tip of that hyphal tube. And in order to keep that tip supplied with stuff, in order to keep organelles, that tip filled with organelles and cytoplasm, there has to be a flow from the interior of the fungus, the interior of the colony, towards those tips. And so we visualize in particular, we're most interested in the nuclei in this flow. And so we use an H1 GFP marker to visualize our nuclei. Okay, and so you see the flow. So flow toward the tips, continual flow toward the tips. And as you go back into the colony, you're feeding more and more tips. They're sort of branching to the left of this image is sort of creating lots and lots and lots of tips. And so you see bigger fluxes of flow toward those tips. But if you go into the deep interior of the colony, things just get crazy. So this is if you look at the flows of the nuclei in the interior of the colony, they're really complicated and multi-directional. Okay.
This flow of nutrients is along the osmotic region?
So it's hard. There is a pressure gradient that drives the flow. It's unclear whether you have to engineer the pressure gradient directly using the osmolytes or just by creating new space. You're varying you create a pressure deficit at the tips. So the problem with trying to achieve an osmolyte balance is, of course, the osmolytes within the fungus are the sugars and the salts that it's absorbed from its environment. So that you're limiting what you can eat if you're using the osmolytes to create the flow. But I mean, the fungus uses about 40% of its genome is devoted to metabolism. So it could very well be that it's engineered into the osmolytes. But I don't know. Nobody knows, actually. It's a good question. So there is a pressure gradient, though. Okay, and so the curiosity that we had, we sort of look at these flows. I don't know how many videos I actually included in this talk. I don't think many. But the thing that I look at, you can see there's a nice analogy between this and cars in a city. You sort of watch these nuclei moving around. It's a lot like watching the headlights of cars flowing around in a city. I mean, not LA, but I mean like a city where the cars move. They sort of they're moving around. And so this is the origin of our analogy, which is, okay, you're seeing things moving around in a city. Are there traffic jams in this city? And so traffic jams, it turns out, are essentially a ubiquitous problem in any transport network. And so we see them in sort of our in the flow of cars around LA, but you also see traffic jams in data networks and things like this. And the problem is that if you make a plot of the speed at which your vehicles go or your data goes as a function of the density of the data or the vehicles, speed always goes down with density. Okay, and so it goes down and down and down until the network gets log jammed. So this is what happens on the 405 essentially every evening. Like we get past the capacity of the highway and just everything jams down. Okay, and so what we were curious about is whether the same process could be seen in fungal networks and these flows of nuclei. And so we made a plot. So we're not very good at experiments, but we are good at analyzing images and data. So if you, this is a plug. If I can plug anything that we're capable of doing that might be useful to you guys, we're very good at image analysis. And so we analyzed the flows of the nuclei, the traffic on these fungal highways as a function of how densely packed the nuclei are on the freeway. And we saw the opposite effect. So don't worry too much about the data that's being shown here. We have groups of nuclei here, and there's another set of groups of nuclei here, and I'll actually show you what these are in a second. But the speed goes up with density. So this is a freeway or a freeway system where the more cars there are on the freeway, the faster everyone is getting to work. So how does that happen? Well, there's a lot of stuff involved in this. But what are the red points here? First of all, where do those come from? Well, if you look at videos of the nuclear flow in the right hyphae, on the right freeways in this network, you actually see that the nuclei bunch up like this. Aha, you say, that's a traffic jam. You said there were no traffic jams. And it's not a traffic jam. So, okay, and these things, not traffic jams, travel a long distance through the network. And then we're gonna skip some of the details of this because I don't want to. Do I have to end at 10:02? Can I go over by like 5 minutes or something?
Yes.
Okay, that's good. You were a little nervous. Can I go over by like 5 hours? Is that? So we're sort of so they're actually, as you saw from the plot that I embedded them on, they go faster than the individual cars. They're not traffic jams. The word that we're using for them is pelotons. That's the term that's used in cycle racing, which is no longer popular in the US. But but you know, back when it was popular, when we actually cared about the Tour de France, you may have noticed that the cyclists tend to form these flocks. They're called pelotons, which is the French word for bullet. And so by forming these flocks together, they actually can slipstream off each other. And by slipstreaming off each other, they can go faster than individual cyclists can. And so there's a cooperativity in cycle racing, and there's a cooperativity in these nuclear flows whereby more nuclei go faster. Okay, and so we won't worry too much about the details of this because I don't want to overrun too much. But as part of the analysis that we did is to actually see why the fungus is able to create faster flows the more dense the traffic is. And it turns out that it's modulating nuclei between two populations. There's a population of nuclei that are actually being trafficked. And so here's a histogram of the proportions of nuclei and their speeds. And there's also a population of nuclei that aren't going anywhere, that are just membrane associated, and they don't have any traffic at all. Okay, so we don't worry too much about the function of these membrane associated nuclei, and we're also not going to worry about the math because, I mean, goodness knows, I'm not going to both overrun and start worrying about PDEs. But we can build a quantitative theory which just uses the fact that there are two populations and nuclei hop between those populations. They hop from being membrane associated to moving with the traffic, and this theory quantitatively predicts the dependence of the speed of the nuclei on the traffic. It predicts how the fungus is able to create faster flow with denser traffic. The problem with this, and this is not just a mathematical abstraction, is that this mechanism is not enough to produce pelotons. So here I am. This is a slide from a math talk. The equations that we use to describe this flow don't have traveling wave solutions. You can't create a group of nuclei and those nuclei will stick together. So we don't worry too much about the details that we did. There's one final factor in this, one final ingredient in this story. And so I said that there were two populations of nuclei and you hop between those two populations. So if we show a plot here, you have nuclei that are moving and nuclei that are attached to the membrane. Well, there are a certain number of attachment sites on the membrane, and you have to encounter one of those attachment sites if you're going to bind to the membrane. So these are just chemical equations, chemical rate equations describing the attachment and detachment processes. And there's one final ingredient that we need, and then just to summarize what we learned, the final ingredient is also that attachment is not a reversible process. So nuclei are moving along, they find a place on the membrane, they attach to that place on the membrane, and then they're released. And if people have questions, I can talk about the analyses that we did. But what we were able to show is that after they're released, the attachment site that they are released from is no longer able to immediately accept other nuclei. So there's a lag time between that attachment site being able to accept a nucleus, losing its last nucleus, and being able to accept the last one. So cooperatively, by having a group of nuclei moving together, they overwhelm all of the surrounding attachment sites, and they're therefore able to move very, very fast through the fungal freeway. Okay, and so we sort of do more math, and so what is the honest thing that we can do with the math that we learn? I mean, we don't want to just make pretty pictures. Well, one of the things that we can do is we can figure out whether we can make a prediction about where we should see these pelotons. Remember, these are fast-moving groups of nuclei, and where we shouldn't see them. And this theory has two parameters in it. One of the parameters is connected to the kinetics of binding. It's the rate at which you bind to a point, the rate at which a flowing nucleus binds to a point on the membrane. And the other parameter, which I'm calling alpha 2 here, is connected to the lag time. It's a measurement of how long an attachment site has to wait before it can accept another nucleus. Okay, and so the important thing here isn't so much the function that these parameters play in the theory. The important thing is how they vary from one hypha to another, one freeway to another freeway. Well, this one here is a lag time. It's based on chemistry. It should be the same from one hypha to another hypha. It's just molecules, and molecules do the same things in the same context. The other parameter is based on the rate at which nuclei find attachment sites, and that depends on speed. The faster you go, the more attachment sites you see. And so, as the fungi which have faster flow are going to have faster binding rates, because each nucleus, as it moves along, and if it's moving faster, cluster will see more and more candidate attachment sites in any given time. So this axis here on the graph is really an axis that represents speed. Okay, and so we can make plots. Oh, it doesn't matter. Let's gloss over this. And so our plot, actually, this line here in this graph represents the boundary between hypha that can have pelotons here and hypha that don't have pelotons. And we find that it agrees with what we're actually able to observe. So we don't know very much about the cell biology, but we can create an honest model that makes an honest prediction. So we don't worry too much about the details, but just to show that faster, larger alpha 1 means that you make pelotons. And so we can test that. Faster hypha should have pelotons, and slower hypha shouldn't. So we can see what happens when a fast hypha plugs into a slow hypha. For example, you start off fast, you start off in the conditions where you should be able to make pelotons, and then you end up slow. So the peloton should go away. And so we're able to engineer that. And so you actually see what happens. We have pelotons in this fast hypha here. It plugs into a thicker hypha down here, and the peloton dissipates, as our theory predicts. And you can also engineer the opposite situation, where you have a slow hypha plugging into a fast hypha. Okay, so why did I go into so much detail about this? Well, I need your help. So what is this good for? Okay, this is a problem. I mean, I'm one of the few mathematicians who cares about this. This is a problem. I built a theory. Is it good for anything? Did we learn anything? And so what's interesting to me, and this is my hypothesis, and I'm so just millimeters away from self-destructing when I make this hypothesis, because I'm a physicist. I don't really know very much about biology, but that my claim is that it's very, very hard to be a fungus. Okay, so now we're going to call for your sympathy to the fungus. It's very, very hard to make one of these networks. Why is it hard to make a network? Well, the network is a cell. It's you. It's what you're made up of. And it's also a transport conduit. And so those two functions are actually a little bit antithetical to each other. And so just to see why that might be the case, if you visualize the microtubules in one of these highways, here we're contrasting two highways. The first one, so this is a beta tubulin GFP tag, and we're seeing the microtubules in two different highways. The top one has very little flow, and the bottom one has plenty of flow. And what you can see is that the microtubules are being continually swept downstream in the lower hypha. Okay, well, I don't know very much about biology, but I know that microtubules are the skeleton of the cell, and so it seems like they might be important. So, and in fact, having them blasted downstream at this high rate might be disruptive to the things that the cell does. Okay, and so this is my hypothesis. My hypothesis is that if we view the system not from the point of view of the pelotons, and this is my last slide, apart from some acknowledgements, if we view the system not from the point of the pelotons, but we instead make a time versus nuclear density plot, where we take a length of hypha and we measure at each time point and at each distance along the hypha the number of nuclei that are present there, what you see is that the passage of pelotons creates phases. So if you stick at some point in the hypha, some distance along the hypha, and the green intensity here represents the number of nuclei that are present, what happens is that the organization of the flow into these fast-flowing pelotons creates phases where there's almost no transport, and then blip, a peloton goes by, all of your transport occurs. And then almost no transport, and blip, another peloton goes by. So you're alternating between phases where you have transport and where you're quiescent. And so this oscillation in time between transport and quiescence, I think, is a way that the fungus is able to engineer an oscillation between its functions. It can do all of its transcription and remodeling during these phases of quiescence, and then it has to do all of its transport, and it concentrates its transport into a very, very short time interval. Okay, so that's my hypothesis. I want to thank the microfluidics lab. We're mainly an undergraduate lab. So not everyone in this picture is undergraduate. I want to especially emphasize Emily Dresser, who is visiting from Connecticut, and also the people whose conversations inspired me to start along this trajectory, and of course my funding sources. So thank you very much for your patience.
Time for questions.
Oh, do I have to take my own? Oh, yes, there we go.
Of course, as biologists, we're always interested in how natural patterns of diversity, variability, can help us test ideas that we might not say about dispersal. And I'm just wondering, there must be mushrooms that have greater power to dispersal than others.
Yeah.
Some are growing up in windy environments, some in very still environments.
Absolutely. So this is one of the...
In terms of their water.
This is a wonderful question. So yeah, we have 15,000 species of fungi. I mean, we've tested this principle only in a couple of species that we can get lots and lots of mushrooms from. You know, so basically the easy cultivars. But it would be very interesting to take this into the field. And so one of the big problems, at least in my research program right now, is that we're able to define these adaptations that the fungus is clearly optimizing for. Like it's clearly trying to do something very, very well. So whenever you sort of look at parameters that the fungus could tune, it does tune those parameters. But the question is how those actually relate to dispersal competitiveness in nature. And I think that that's sort of a leap that we desperately need to make, but we're not capable of making it yet. In terms of doing like a sort of interspecies comparison, that's an excellent next step. So we could look at different mushrooms and different environments. We just need to collect the mushrooms and do that. But that's absolutely a great idea.
Beautiful talk. Have you thought yet about how the morphology of the mushroom caps influences the fluid dynamics and the turbulence?
So this is a really good like we've thought about it, but only in a very bad way. So in a sort of a very sloppy way, in the sense of like we know when we're looking for the gravity current to look for a part with a part of the mushroom cap that's a little bit fluted, because there's this if you look at the dependence of velocity on thickness. Velocity goes up the thicker the gap between the mushroom and the ground is. So by deliberately creating flutes, the mushroom, so parts where the cap goes up, the hymenium lifts, the mushroom is able to direct where its gravity current should go. But we haven't really like looked to see whether that's correlated with where it will be an advantageous direction for the mushroom cap to go. So this is it's a really good next direction to go in. I mean, again, we have these 15,000 species with like huge morphological, 15,000 that we actually have been named with huge morphological diversity. So, question.
You mentioned that the mushrooms lose water pretty fast. Do they have adaptations to prevent water loss through the top of the cap and only lose water through the gills, or do they just lose it sort of everywhere?
So we're able to, we've looked to see whether the mushroom is directing its water loss. So basically we just cover different surfaces within the mushroom with petroleum jelly to to create a sort of water impermeable barrier. And it looks like the mushroom loses water from every which surface. And it's a little difficult to sort of, in fact, if you cover any surface, the mushroom continues to lose water at the same rate, because evaporation has a peculiar dynamic. It doesn't depend on surface area, it actually depends on your local water vapor atmosphere that you create. So essentially, if you block water vapor loss, you don't affect any of the dynamics in the system. If you like slather the top of the cap with petroleum jelly or something like that. But it could be that the fungus is trying to conserve water in some other way, at least like water loss from the stalk or something that's not connected to the function of dispersal.
Other questions?
I have one more.
Oh, one more.
So your hypothesis at the end about the phase shifting between the two different purposes, I have no instinct for the kinetics of any of the things that fungi do, but is 100 seconds really long enough to do all your dynamic remodeling before another proton comes through and wants to do?
Well, we're essentially asking how long do you need, that's a really good question. I mean, how long do you need to transcribe a protein, for example? And it's certainly long enough to do that. It's also long enough that you can do, like this is one of the nice things about working with fungi is, the yeast community. Like those are one of the wonderful, I mean, they think they're studying, human biology, but really, oh God, there's some video I should be. So this is so a lot is known about how like endo and exocytosis work, and so we know all of the timescales associated with those processes are like short enough that you could pack them in. Whether you could pack enough of them in is like a big mystery in this. So that's the last thing. I think I would be much more comfortable if I had a way of testing this speculation. Like if I could actually interrogate whether the nuclei were doing anything in the interphases. Like if we had some way of labeling the transcription factors certainly we can label transcription factors. The problem is because the environment is so dynamic, like sort of everything is being blasted downstream. I mean, even visualizing something as ubiquitous as microtubules is really challenging, and finding a transcription factor is beyond what we can do right now. But we're very short of actually having any kind of test of the hypothesis that I made.
I was wondering, it looks like ambient conditions such as relative humidity or vapor pressure deficit of the air is a very important factor for the distance of dispersal. And so how you could bring this in your model?
So it's definitely something that we can alter in the lab as a variable. So our delta rho is something that we have a fairly good control of because it comes from physics, and that trammels up the ingredients, and that include the relative humidity, which affects the evaporation rate and things like this. How you then translate that to the field, which would be the dream of actually saying this is ecologically meaningful, that the fungus isn't just capable of doing this, but it's actually, that's what it's doing in nature. That's very challenging, and all we have is the indirect evidence that sort of fungi grow in ways that conventional modes of dispersal that don't involve creating your own wind would make very challenging. But yeah, it would be great to like actually go, you can see, I guess a lot of you are sort of field scientists, and so it must seem very naive, but wouldn't it be great to actually do an experiment in nature? Wouldn't that be amazing? Yeah, we've never done one, but yeah, absolutely. It would be very good to do those correlations, and especially to also figure out how climate and weather pattern changes would affect, because all of these things that you're talking about depend on climate, and so this is sort of, like definitely connected to issues of global change and rain shifts and things like this. Where can you actually use these mechanisms to disperse?
And also, are you planning to do something for underground? How they move, I suppose, underground?
Underground. So what do you have in mind? Like the hypergeus mushroom?
How they suppose move?
How, so the mushroom spores move?
Yes.
So I mean
There
I guess I don't know exactly which species you're thinking of. If you're thinking of like things like truffles, the tuberalis, so then there's sort of people, I mean the general idea there is that they're sort of dispersed by animals, and they use chemoattractants to attract the animals. The curious story for me, and sort of part of why we started working on this, was that I moved to California, to Southern California, and I couldn't find mushrooms anymore. I don't know if anyone sort of had this experience. Yeah, well, you're wandering around, and then you like go out with someone who knows the local flora, and they tell you, oh, you have to look for mushrumps. I don't know if you know about this. So basically, a lot of mushrooms here, they stay under the leaf litter layer. So you have to like be looking for like little lumps of, so like, and then you ask them, how on earth are they dispersing their spores if they're under the leaf litter layer? Well, our mechanism works for that because that's a crowded environment that is good. But actually, the general prevailing view right now is that they're insect dispersed. So you have the same species in Northern California being wind dispersed and insect dispersed here. So that I felt like was one of the reasons we had to sort of start investigating whether crowding you could still disperse. But that's not exactly the same as being under the ground. It's just under the leaf litter layer.
Okay, it's time to go. Thank Marcus again.
Thanks.
Really delightful talk, actually.
Yeah.